Welcome to our first lecture video on the topic of humans in the world of biology. This is in chapter 1 of your textbook, is where you'll find the information in this PowerPoint. So when we look at human biology, we're basically studying the world of life. So when we look at biology as a subject, that would include plants and animals and all the other living organisms, but obviously human biology is studying the biology specifically related to humans. So biology as a general term, however, just means the study of life. Bio means life. So we're going to talk about a couple of different major topics as it relates to human biology and getting started. We're going to talk about some characteristics that all living things share, and talk about levels of organization, a little bit about the scientific method, and then how we look at scientific claims and evaluate their accuracy. So first of all, looking at basic characteristics of all living things, there's a couple of statements we can make as it relates to living things. And all things are composed of 4 basic building blocks. So nucleic acids, maybe you've heard of DNA and RNA before, proteins, carbohydrates, and lipids. We're going to talk about each of these organic molecules individually, but when we look at the human body, it's essentially a combination of all 4 of these organic molecules. So our first characteristic of life is that living things contain these 4 organic molecules, and how those organic molecules are arranged can be different from organism to organism and even within an organism. So we'll be talking a lot about these structures throughout the semester. So looking at another characteristic of living things is that living things are composed of cells. So the cells are the basic unit of life. So they are the smallest units of life. They come from pre-existing cells, and they're able to grow in size and in number. So when you look at the human body, for example, and all the different things that the human body is capable of, it's because of the function that occurs within individual cells. And we're going to study the structures of the cell in next week's week 2 topic. And cells use the energy and raw materials that we bring into our body to carry out metabolism. And metabolism is simply defined as all the chemical reactions that occur within our body to keep us alive. So the second characteristic of life, then, in summary, is that all living things are composed of cells. A third characteristic of life is that living things grow and reproduce. So as we all know, we started out as one cell. When sperm and egg united from our father and mother, it formed one cell called a zygote, and then that cell divided many times to become a human being and a fetus that then is able to live outside the womb. Living things use energy and raw materials, so living things have to acquire energy from the outside. Plants acquire energy from the sunlight and convert that to sugar or glucose, but humans have to take their energy from other living things. So we take in our energy in the form of food, and that is the raw material that we use to fuel the chemical reactions in our body. All living things respond to their environment, and that means that we have to react to the environment around us. Sometimes there's conditions in the environment that need us to respond in order to stay safe or to just maintain body temperature. For example, when it's cold outside, we wear a jacket. When it's hot outside, we take our jacket off, and that maintains the environment inside of our bodies to keep us at the right temperature. So here we can see this chameleon is responding to the environment. It sees food, so it takes it in via its long, sticky tongue. The same thing with us. When we have the need to eat, we feel that urge, and we look into our environment for something to eat. So that's important that we respond to our environment. And then living things maintain homeostasis. That's a key concept of a class like human biology. Homeostasis is simply described as the fairly constant internal environment that we maintain. So when we say fairly constant, we mean that, there is a little bit of fluctuation. So our body temperature might go up a few degrees or down a few degrees depending on the outside temperature, but it's fairly constant. The same thing with our heart rate, our blood sugar, our blood pressure, all those things that keep us going and keep us maintaining life have to be maintained within a specific range. So this concept of homeostasis is a really important concept, and it's part of that response concept we just talked about, that all living things respond to their environment. If we didn't respond, we wouldn't be able to maintain homeostasis. For example, if I put some plants in the window in the springtime, I'll find that those plants will grow toward the sunlight. That's an example of living things responding to their environment. That is a way that those plants can maximize photosynthesis and maintain the energy they need to keep growing. So just the fact that plants grow toward the sunlight is a perfect example of how living things respond to their environment, as well as how they maintain homeostasis. So another concept is looking at evolution and how populations will evolve and adapt to their environment over time. So when we look at, for example, we look at orchids living in a tropical forest environment, they can climb up and coordinate with other plants in the environment to make sure they get enough sunlight. So they can kind of branch up those trees, and they are supported by the branches of the trees to make sure that they're exposed to the sunlight. So that's another way that we have been able to change or evolve to make sure we can maintain homeostasis and get our needs met. So when you look at evolution, evolution is defined as change over time. We know that humans share a lot of characteristics with other animals, especially the vertebrates, particularly mammals. So we are most like those that produce live young and nurse their young. That's how mammals are defined. And humans are in that group of living organisms. So we have evolved, just like other animals have evolved over time, to adapt to our environment. And then we also can look at all living things and organize them into specific domains and kingdoms, and we'll talk about some of those here on the next slide. So when you look at living things, we can group them into three major groups. The domain bacteria, which are one-celled organisms that we're familiar with. They cause infection in our bodies if we don't keep ourselves clean, or there's an injury that might occur, that we have a break in the skin and bacteria in the environment enter. But we also have healthy bacteria, for example, in our gut and on our skin, that keeps us infection free from the bad bacteria. We also use the bacteria in the environment for some of our food processes. For example, we use the bacteria lactobacillus in our yogurt and cheese products. So some bacteria are healthy, some are not healthy, and some are normal flora, like I said, in our digestive tract and on our skin. Another major domain of living things are the Archaea. Archaea live in extreme environments, and they are not something that we come in contact with too often. We see them like in extreme, like sulfur beds and the vents in the ocean. But they are an organism. They are also one-celled. They lack a nucleus, just like bacteria. But we fall into the domain of eukarya. Eukarya are all the living things that are much more advanced and developed, and all these organisms have cells with a nucleus. So eukarya includes the plants, animals, protists, all the living things that are around us are in the domain eukarya, and that includes us. We fall into the kingdom Animalia because we are more evolved, say, than plants, fungi, and protists. But we're not going to spend a lot of time looking at the other organisms because this is a human biology course. We're going to focus primarily on humans. So what makes us unique in terms of evolution is that we have a very large brain relative to the size of our body. Because of that, we're able to use advanced thinking skills, critical thinking, problem solving, communication. We're very social organisms, and that makes us unique from the other animals that we evolved with. So we are able to stand up on two legs. That's obviously another distinct feature, that we can stand completely upright. And again, we have a culture and social influences and beliefs and behaviors that are unique to our species. So when we look at how life is organized, we can look at the most simplest piece that we are made of, and that is atoms. Atoms are the most simplest unit of matter that makes up what we call molecules. So when two atoms of hydrogen come together with one atom of oxygen, that forms a water molecule here. And then when molecules come together for a specific function, that forms a cell. And when cells work together, for example, the cells of our muscles work together to contract and move our body, that's a tissue. So muscle is a tissue made up of many muscle cells. When different tissues come together, they form an organ. So we know that the heart has 3 layers of tissue that make up the layers of the heart wall. We'll talk about that when we get into the cardiovascular system. And different organs working together form organ systems. So the heart and the blood vessels together make up the cardiovascular system. And all the systems working together, for example, the respiratory system, the cardiovascular system, all of the systems working together form an organism. So know these in order, from the simplest to the most complex, and be able to list them in order. So then we can organize a group of organisms together that are living in the same environment. We would call them a population. For example, a group of fish living in a pond that are all the same species, like for example trout, we would call that a population of trout. They're the same species living in the same location in a pond or a river. And when we look at a community is made up of all different species living in a specific environment. So if I looked at that same pond and I looked at the frogs and the turtles and the fish and the plants, that would be a community of species because they're made up of different living things in the same location. And if I take that community and I add that physical environment, for example, I factor in the temperature, and I factor in the water, and I factor in the soil, some of those things that are non-living, that are part of the physical environment, that I would call an ecosystem. So for example, a wetland is an example of an ecosystem. It consists of the water and the soil around the wetland, and it consists of all the living organisms within there as well. And then if I look at all the ecosystems of the world, that they are clustered into specific biospheres. And we have several different biospheres scattered around the earth. And for example, the ocean is the largest biosphere that exists on earth, but there's several others as well, and we'll get to that at the end of the semester. So when we look and approach science, especially in health and human services or an educational environment, we're going to be exposed to lots and lots of scientific claims and ideas, and we have to sort through that and determine what is truth, what is fact, and what is opinion. So we have this wonderful way of approaching new ideas or observations called the scientific method. And I'm sure you've heard of this before in your previous science classes in high school or middle school. But it's just a systematic way of making sure that we look at an observation or some claim that's being made, maybe on the internet or in the newspaper, and we're trying to determine if it's objective or subjective observations. Objective means that everybody would agree. For example, if I look at a plant and I say that it is 6 inches tall, everybody would agree, if they had a measuring tool, that the plant is indeed 6 inches tall. That's not something that is opinion based. So we say that's objective data, something that can be directly measured. Subjective data would be that you have a group of students, and if you use a particular teaching method, the teacher reports that students enjoyed the lesson. Well, that's a subjective piece of data. You can't measure enjoyment. So that would be more opinion based. So it's really important when we're gathering data that data is as objective as possible, which means fact based, something that can be easily measured and not subject to opinion. If it's opinion based, we say it's subjective data. And we have to be really careful when we gather our information, because the whole purpose of the scientific method is to help us make decisions for not only our own personal well-being. For example, if you're looking at the latest claims from a fad diet, or whether or not to vaccinate your child, or the well-being of the earth. For example, if you're looking at, should I allow a frack mining factory to come into my community, or should we allow recycling, or should I allow oil drilling in our community? So those are all things that affect the well-being of the earth as well, and knowing how to separate subjective versus objective data using and understanding the scientific method is really good for like I said, for the well-being of yourself personally as well as the well-being of the earth. So when you look at the scientific method, it's really, really important that we make our observations, ask questions about those observations, then make a prediction to start the experiment. And that's what a hypothesis is. It's a predictive statement. It usually starts with the letter or with the word if. For example, if students visit the tutor at least 2 hours a week, then they will get a score of a C or better on the next exam. That's a predictive statement. It's measurable, it's observable, and it's making a prediction of what I think is going to happen. So that's the key thing, is starting with a hypothesis where you make a predictive statement. And again, usually it starts with the word if. So then we would set up two groups of people, a control and experimental group. The control group is one that wouldn't visit the tutor. The experimental group would be the group that does visit the tutor. So if I find that the experimental group does better than the control group, then I could say that yes, the tutoring helped students do better on the test. But if I found, but if I found that the control group and the experimental group had the same score on the test, then I know that my experiment failed and that my prediction that seeing the tutor would make a difference was incorrect. So it's really important to have two groups of people, one that is exposed to the experimental variable, which is the experimental group, and the control group that is not exposed to the variable. For example, this would be the group that would not receive the tutoring. And from that information then, I would reach a conclusion. Looking at the scores of both groups, I could say, did tutoring make a difference? And then I might want to ask more questions, like if tutoring did make a difference, well, how much tutoring? Or what type of lessons were covered in the tutoring session? And then the really important piece of the scientific method is it has to be repeatable. And I can't emphasize that enough. When we're looking at science, if it's not repeatable, if I can't take that same experiment and get similar results using the same steps, then something's wrong. There has been many scientific claims. For example, the one that was developed many years ago saying that vaccines cause autism, that was not repeatable. But because the information was spread around the world from that one person's experiment, many people tacked on and believed that vaccines did indeed cause autism. But when scientists tried to repeat the experiment, they could not find any data to support that vaccines caused autism. As a matter of fact, that particular doctor lost his license because of the data he put out was faulty. We make up a predictive statement, which is a hypothesis of what we think will happen, and then we go ahead and run that through our experiment and data process. So it's important when we look at this flow chart here, we make the hypothesis that eating oatmeal lowers blood cholesterol levels, and then we make a prediction. So we say that if oatmeal consumption lowers blood cholesterol levels, then a person's cholesterol level will be lowered by eating a bowl of oatmeal a day for 6 weeks. Then we would perform the experiment. So the information we gather to support that prediction is called data. So data, again, should be objective, not subjective. And then we would want to have two groups. We'd want to have the experimental group that eats the oatmeal and the control group that does not eat the oatmeal. And if this were a medicine, let's say we're going to say that a particular blood pressure medicine or a particular cholesterol lowering medicine works better than another, sometimes we don't want the control group to think they are the control group. We don't want either group to know who's experimental and who's control. So we might give the control group a placebo. And what a placebo is that's a pill that does not contain any medicine in it. It might just be simply a little bit of a sugar pill or some other benign, inert substance that is not the actual medicine that's being tested. That's really important in clinical trials when we're looking at new medicine and their development. We're going to gather two groups of people. One group is going to get a placebo, which does not have any medicine in it, and the experimental group would get the true medicine. Like in this case, it would be a cholesterol-lowering medicine. And then we would compare the cholesterol levels of both groups after a period of time. So again, if I find that the cholesterol levels were the same between both groups, then I would say the medicine didn't work, because obviously the control group should have had no change in their cholesterol level. For example, if it was high, it would remain high. And that would mean that the medicine did not work if both results from both groups are the same. So looking at the data here, we can look at the person that was eating oatmeal. If I look at the triangle here that shows 2 ounces of oatmeal versus 3 ounces here versus 1 ounce in this graph, and then versus farina, which is another type of a grain that one can eat for breakfast. This is what we see, for example, in malto meal. I can see there's definitely differences here, which showed the steepest drop in cholesterol. So here's my cholesterol levels on the side. This is the dependent variable. This is what we're looking to change in our experiment. And this is time. Time usually goes across the bottom, across the horizontal axis. So we have the vertical axis, which has my variable, my experimental variable, which is the cholesterol levels, and then the time across the bottom. So here I can see the steepest drop occurred for those that had 3 ounces of oatmeal maybe in the morning or for dinner, or whatever, once a day. So we can see that there's a specific amount of oatmeal that's recommended to get the highest drop in cholesterol levels. And we can also see that it did drop with the other amounts of oatmeal as well, but in this case with the farina it actually went up a little bit, so we know that farina would not be a good choice for lowering cholesterol levels. So when we look at how we tackle a process, tackle a question, or tackle an experiment, sometimes we can gather enough information by looking at a lot of smaller pieces of data. For example, we call this inductive reasoning, is when we take numerous facts and then develop a conclusion or develop a hypothesis based on lots of different pieces of data. So that's called inductive reasoning. So you're starting with small pieces of information and making a statement from that. The other type of reasoning in approaching a scientific problem is making a statement, a general statement, and then looking for data to support that statement. So we're looking at more of a general statement and then looking at specific conclusions from that statement. So deductive is starting with something bigger and then breaking it into pieces, where inductive reasoning, on the other hand, is looking at the pieces and coming up with some general statement from that. So it's 2 ways of approaching a problem, and it depends on the experiment of which is more appropriate. So when you look at the scientific method, particularly looking with medicines or things that are used by humans, we have to have some type of test subject. And of course, we wouldn't want to expose, a human being to a harsh chemical that might be bad for the skin, say a new cleaning agent. So we typically use animals for experimentation and testing. And rodents are a good starting place for testing the effects of certain chemicals or medicines on living cells. So we start with that and then we move upward from there. So that's what we call clinical trials, is when we start using humans as the test subjects rather than animals. So we sometimes see this in cancer treatments. Patients will maybe not be responding to the typical treatment regimen regimen for their cancer, so they might go to medicines that are being used in clinical trials. But those have not been fully FDA approved yet, but they're in the experimentation process. But in patients that don't really have any other options, they're comfortable with going to clinical trials, because if they don't treat the cancer, it's going to take their life anyway. So many people will enter the clinical trial process just to have some hope for treatment and recovery and remission. So sometimes we enter into testing. College students are sometimes recruited to enter clinical trials for certain medicines, and they'll enter what's called a double-blind study. And what a double-blind study does is it tells the researchers, and it is set up by an outside agency where the researchers and the participants do not know which group is receiving the experimental treatment. And it really is good for making sure that there is no bias in the group and in the experiment. So when we look at groups of, say, populations, and we're looking at a particular disease, for example, we can perform what's called epidemiological studies. Epidemiology is the study of populations and diseases and health conditions that occur within populations. For example, if we're looking at the AIDS virus across the world, we know that there are certain countries that are at higher risk for AIDS than others. For example, Africa has a high risk for AIDS, particularly among women, and that's the biggest group of new AIDS patients. And it's still happening today for a number of different factors. So there's lots of different methods in which we use the scientific method to study humans and to benefit humans. So again, it could be the use of a new drug, where the experimental group receives the drug, the control group receives a placebo that looks like the drug, but there's actually no medicine in it. Sometimes we perform double-blind studies where researchers and participants don't know who is receiving the treatment. That really eliminates any bias that might happen during the experiment, and it's very helpful to prevent, falsifying data or people with hidden agendas trying to force their idea into the scientific method. And again, looking at epidemiology, and this is really an important one for people going into social work, human services, or education, is just understanding the patterns that exist in specific populations and some of the factors that lead to those patterns. And sometimes it's not simply physiology or anatomy. There's a lot of culture and economics and even politics that factor into some of the diseases and health challenges we see in different populations of humans. So when we look at clinical trials, here's an example of how a new drug comes on the market. It enters into, first of all, just being tested on animals, and if it's considered safe for animals, then it enters into clinical trials, which again is for human use. So phase 1 is just looking at, is it safe for humans? Are there serious side effects of the drug? Then phase 2 would be, does it actually do what it's supposed to do? For example, if it's a blood pressure medication, does it actually lower blood pressure? And then as it enters phase 3 is, how does it do compared to the other blood pressure medications? Is it better, or is it a comparable? Does it have any benefits compared to what's already out there? Because pharmaceutical companies are really interested in the data resulting from clinical trials, and if the drug is no better than what's already out there, they're not going to invest the money in the marketing and packaging of these drugs. So it has to really be better than what's out there, or at least an alternative to people that maybe have a sensitivity or an allergy to one type of medicine. Maybe they need a new medicine. So once it passes phase 3, then it's on the market and can receive that FDA approval. So when we look at how to evaluate scientific claims, it's really, really important we understand what good information is and what resources we can rely on. So much information right now is being delivered over the internet, over the web, and that is dangerous, especially look at Facebook, and maybe a good friend of yours posts an article that came out stating that a particular food is really bad for you, or a particular food is amazing and it's a superfood and we should eat a lot of it. We really have to be careful with that and make sure that those sources come from scientific research, not just a group of people saying, hey, I drank this and my pain went away, or I had a ton of energy. We really have to be careful with that because, for one, everybody's different, and most importantly, if it's not backed up by scientific data, it's not something that we want to spend a lot of time and money on until we've determined that it is truly doing what people say that it's doing. And the vaccine and autism debate is one that is really a perfect example of poor scientific backing to show that vaccines are bad. So we call that correlation without causation. So for example, if we look at autism and vaccines, we know that babies receive their MMR vaccine at a year old, and babies also will be diagnosed with autism a little after the 1-year visit. And that can be related to the fact that babies aren't diagnosed with autism until the 1-year visit. Doctors often will wait to see if behaviors or tendencies resolve by the 1-year visit or improve before they'll give the diagnosis of autism. And they also give the MMR vaccine at the 1-year visit. So just because both of those are happening at the same time doesn't mean that one causes the other. So we call that correlation, which means MMR vaccine and autism diagnosis are correlated because they happen at the same time, at the same visit, but it doesn't mean one causes the other. So that's where we really have to be careful that where we find our data. So good sources of data are websites that end in, for example, .edu, .gov for government, .org for some organization that's usually funded and has a lot of scientific research behind it. Those are good places to go to get information regarding a particular claim. But if it's simply a .com website, that's not a reliable source of information. .com websites can be put out there by anybody, and we really have to be careful with that. So .edu, .gov, .org, those are all good sources of information, but .com should really be looked at with some scrutiny. So now, after this chapter, you should understand what the basic characteristics of all living things are. We kind of went through that list, and we did some classifying of atoms to molecules to cells, all the way up to the level of biosphere. That's the levels of organization. And we also talked about the importance of the scientific method and the importance of using critical thinking to evaluate scientific claims. So that concludes today's lecture on Chapter 1. Go ahead and get started on the homework for Chapter 1, and we'll see you next time.